The last thing that we're going to talk about in this chapter is a application of the cell cycle that will unfortunately probably affect all of our lives directly or indirectly, and that is through cancer. Cancer is a malfunction of the checkpoints of the cell cycle. So in the cell cycle, we have these checkpoints to say, hey, do we have enough resources in order for cell growth to continue? Hey, is our DNA looking okay? And in metaphase, we have a checkpoint that says, hey, are all the spindle fibers lined up properly? Are we attached all the chromosomes in the right way? Has everybody buckled their seatbelt in? Okay, so if we are, all right, then we can move along. And it's a failure in one or more of these checkpoints that can lead to cancerous cells. Okay, so mutations are permanent changes in the sequence of DNA. So we might see differences in the sequence of A, C's, G's, and T's in our genetic material. And most of the time, even if we do have like a little whoopsie-doopsie mistake, that's not going to affect the protein. So we call these silent mutations, because even if there is a mistake, we're probably going to make the protein that will still do the job that it's supposed to do. Occasionally, though, we might have a mistake that leads to a protein that is going to do something that we don't want it to do. Right? We're going to have this abnormal protein that's going to do a different function. Or it might fail to produce a protein altogether. And so then we're lacking a regulator or an enzyme or something like this that can be very, very detrimental to what we're doing. Okay. And we have all of these rigorous checks and these checkpoints, and we have this DNA proofreading mechanisms that we're going to talk about when we get to the DNA chapter. And yet, even though we have all this redundancy in checking to make sure that everything's cool, there's still going to be mistakes that slip through, because that's just how mistakes be, right? And those small mistakes are going to accumulate over time. So the more times that your cells replicate, you're more likely to have mutations, which is why older folks are more at risk of cancer. Okay. So in the cell cycle, we're going to have these special proteins that our jobs are to be, hold up, let's check everything out. Let's do some quality control here before we move along. Okay. And so these proto-oncogenes, these are genes that we find in the DNA that produce proteins that are going to encourage the cell cycle to move along. Okay. If there's a mutation in one of these proto-oncogenes, then we will call it an carcinogen, right? Because that protein's not going to function properly anymore. And so it can potentially cause cancer. So if the protein is saying, yep, go ahead, when it should be saying, wait, hold up, stop for a minute, right? Then that's going to call this continual, let's go, is going to cause cancer. Okay. We also have cancer suppression genes. So these genes are going to stop the cell cycle at a checkpoint. And if those are malfunctioning, we're not going to have an off switch in order to say stop the cell reproduction. So either our stop or our go gets messed up, we're going to potentially lead to cancer. P53 protein job is to check to make sure that there's no DNA damage. We're checking to make sure that everything in the cell cycle is doing what it's supposed to do. We're checking to make sure that we have enough oxygen so we're not going to cause any more mutations. Now, normally, this P53 says, okay, wait, stop, hold up. Can we fix this DNA? Okay, yes, we can. Okay, then let's go ahead and continue with the cell cycle after the DNA is all fixed up. If the P53 says stop, hold up, let's check to see if we can repair this. And we cannot repair the DNA, it's too far gone. Over there to the recycling pile with you. We're going to do apoptosis, which is programmed cell death. We're going to recycle you and use your stuff to make proper cells. Okay. When we are seeing this damaged P53 protein, it's not going to say stop, hold up. We're just going to keep shuffling through, keep going with the cell cycle. Okay, again, about 50% of human cancers are due to a mutation in this P53 gene. Okay, and what it does, one of the functions of the P53 gene is to signal time for apoptosis, which is programmed cell death. This is the process, and we're going to pull the cytoskeleton away from the cytoplasm. We're going to get this kind of funky membrane blebbing. It's going to start to fall apart, lose its structure, until finally it turns into a pile of mush that we can then use to regenerate new cells, right? The RB protein is another cell growth regulator. So if we have failures on those parts, and these RB proteins occur at each of these checkpoints. So if there's a failure in the RB protein, then we can be in big trouble as far as cancer developing. Okay, now tumors require a lot of nutrients and oxygen, and so what they tend to do is that if we have a tumor, it's going to activate and provide signals that say, hey, blood vessel, I need more food over here. Why don't you create a new branch, right? Create a new side street, and so you can come over here and feed me. Okay. So if the tumor gets to a point where it's large enough, it might even enter the bloodstream. So we might have this cancerous cell break off from the tumor, get into the bloodstream, and travel to a new area and attach onto a new location. And at that point, it's really hard to control the cancers, and it's a lot harder to treat that. So if something feels wrong with you, go get it checked out. Listen to your body. Make sure that you're getting your proper preventative checkups, so that you can catch these when they're small and when they're easier to treat.